Hi everyone, welcome to the WebEVTalk series. Today is the 21st April 2020, and it's my pleasure to welcome Associate Professor Rebecca Lim. Rebecca Lim is a Deputy Center Head of the Ritchie Centre, Monash University. She is an expert in amniotic stem cell therapies and have been successfully leading 7 competitive grants, and she also received prestigious NHMRC Excellence Awards in Australia. Her research is focused on translating discoveries around regenerative medicine and cell therapies to clinically useful outcomes. Beck has successfully brought her basic research to 2 registered cell therapy clinical trials. In 2018, she translated her baby have no other effective. Her experience in implementing basic research to clinical translation, and her current work in extracellular vesicles in regenerative medicine. Please welcome Rebecca Lim.
I'm with the Department of Obstetrics and Gynecology at Monash University, and I also have a dual appointment with the Ritchie Centre at the Hudson Institute of Medical Research. Gina, could you just flick over to the next slide, please? So what I'll be presenting on today is to give you a little bit of I guess, an understanding as to our facility, how we've gotten to where we've gotten. I believe in large part this might actually be an environmental thing. Being a discovery scientist, not having any medical training, I did not at any point in time set my goals to I will enter the clinic, but was very much through a series of mentorship and a little bit of the culture of the research environment that I'm in. And so the history of perinatal stem cells, I thought I'd quickly introduce as well, because it might sound a little bit unusual for perinatal stem cells to be used in regenerative medicine, and how we've moved from our early discoveries into cell-based therapies and clinical trials. I'll also be covering a bit about our preclinical journey, how we went about selecting animal models, what was deemed appropriate at the time, who our clinical champions were and the role that those clinical champions played in the translation of our research, how we then went about developing potency assays and clinical release criteria for our cell-based therapies, and what we went through, the process that we went through with our first-in-human clinical trials with the cell therapy, and then how we can then take those learnings into EVs. And hopefully some of the pain, possibly agony that I've gone through in the past decade can actually help you who are listening in looking to move into EV therapy, save you a bit of time and a bit of agony on your part. And some tips that I've learned over the years and have been passed on to me by some real experts in the field. So on to my next slide. So what you'll see here on the next slide, Gina, this is a picture of our precinct. And so the brown and gray building that you see smack bang in the middle of that picture is our translational research facility. And this was a purpose-built facility within one of Victoria's largest medical precincts. So we're based at the Monash Health Translational Precinct, and Monash Health is our state's largest public health service provider. And what that means is that it co-locates clinicians, patient advocacy groups, patients themselves, their families, as well as researchers on the one precinct. And what they've done in the design of this building is actually to create natural hotspots. So you'd have a cafe in the lobby where the clinicians, the patients, and the researchers would be interacting with. Often you can't really get to your own lab or your office without bumping into one of these people that eventually, you know, over time you learn that they're just your colleagues. If you just animate the next bit, Gina, thank you. What you'll see here is the back of the building, which allows me to just kind of label where we are. So on level 5 is our research facility. So we've got labs on one end and office space on the other. And the level below that is actually the cell therapy platform where we do the cell manufacturing for our cell-based clinical trials. Level 3 is a clinical trial center where our ambulatory patients are located, and we're physically linked to the rest of the hospital through a bridge. And that becomes important because, as I mentioned, Department of ONG and perinatal stem cells, the operating theaters are where we actually collect our tissues from in order to isolate the cells and from those cells derive those EVs. Thanks, Gina. And so the Ritchie Centre, as mentioned before, is on this medical precinct. And I just had a bunch of logos on here to show you the different identities of partners that are on this precinct. And if you animate on, Gina, what you find is that the Ritchie Centre is actually the academic research center for our Department of Pediatrics and Department of Obstetrics and Gynecology. So perhaps this then makes it a little bit more obvious as to why we've gone the way we have and the cell type that we've gone with. Next slide, please. So the amnion, well, the placenta, but the amnion has had a long history of use in medicine and in surgery. So the very first record of the amnion being used in medicine was actually in 1910. So we've had well over a century's history of its use in medicine. Initially to use as biological bandages to cover up skin burns and grafts. But the ophthalmology field has continued to pursue this, and they're used in corneal ulcer repairs. And if you just animate the next bit, Gina. And perhaps if you consider how invasive pregnancy is in humans, that invasive nature of the maternal-fetal interface during pregnancy could give you some understanding as to why the placental tissue, the amniotic cells, are so immunomodulatory. So in order to have a healthy term pregnancy, a healthy baby at the end of that pregnancy, the mother's immune system must necessarily adapt to the fetal antigens. And as you can see on that top left-hand corner, the placental tissue actually quite invasively enters the maternal tissue. So there's a lot of immunopriming and a lot of modulation of the mom's immune system throughout the course of pregnancy. And what we do know is that when there's a failure to adapt to pregnancy, where the fetal antigens are presented to the mother without sufficient immune modulation, is that you get like recurrent miscarriages, spontaneous abortions, preeclampsia, and the like. So we know that this is a key part of what the placenta does, and of the placenta, the amniotic membrane is what we're interested in. And next slide, please, Gina. So with the perinatal stem cells, you know, the placenta being so rich, if you like, for lack of a better word, with stem cells and stem-like cells, a number of different research groups have gone on to try and understand what cell types can be isolated. Gina, more than being my teleprompter, her own PhD was actually in placenta-derived stem cells. There are a number of different colleagues that are tuned in right now who are working on MSCs from the umbilical cord, as well as the stem cells and stem-like cells from the fetal membranes. And our own research, if you just animate that, Gina, is the amniotic epithelial cells. And the next thing to keep in mind with the amniotic epithelial cells that they have stem-like markers. So they have stem cell markers, traditional stem cell markers like OCT4, NANOG, SSEA4. However, they're not stem cells per se. So they express very high levels of EpCAM. They are epithelial in origin. They do not look like MSCs and would not in culture be mistaken for as MSCs. They're heterogeneous. So if you isolate the cells from the amniotic membrane, you'll find that much like EVs, you will see a diverse population of cells, even though they all came from the membrane, isolated in the same manner, and without passaging already present as being heterogeneous. Some of them will adhere to plastic, some will be loosely adherent, and some can be cultured as non-adherent cells in suspension. And they do not expand well in culture. Next slide, please. So this is one of my colleagues, Francisco. I'm not sure if he's on the call, but some of those on the call will recognize his name. So this is actually from his paper. So it's not just our work alone, but other colleagues around the world have shown that the HAACs do not expand well in culture. This becomes an issue if you're trying to expand cells to get conditioned media to make your therapeutic EVs. So that's something to keep in mind. one of the other things, though, on the flip side is that you can isolate a huge number of cells from a single placenta. So while you can't expand them, you don't actually need the 6 to 8 weeks to get the number of cells that you need, because you get 150 to 250 million cells from a term placenta within a 6-hour isolation protocol. So from a therapeutic point of view, you have a little bit of balance. So they don't passage well, but you also don't need to passage them. However, you do need to isolate from new donors every single time once you run out of a bank. They do have similar characteristics to mesenchymal stem or stromal cells. And as mentioned before, because of the role that they play in pregnancy, perhaps it's not so surprising that they have these sorts of characteristics. I've just listed them on there. We've published on this area, and I'm sure you're not here to really learn about stem cells, but the EV therapeutic. So I've just listed them on there for your reference. Next slide, please, Gina. So the disease that we're interested in is bronchopulmonary disease. It's not the only disease that we're interested in, and I'll talk to you about some of the other clinical trials that we've worked on and are in the process of delivering. But bronchopulmonary dysplasia, or BPD, was pretty much our first in-human trial. And, you know, as I mentioned earlier, because we've got this very close relationship with the neonatologists and with the obstetricians, we're able to then kind of say, well, this woman is at high risk of delivering her baby at 23, 24 weeks. Let's have a chat with her then. Consider, you know, sort of give the patients as much time as they would like to consider going to cell-based therapies should their child then develop bronchopulmonary dysplasia. So BPD is the most common chronic lung disease that you'll see in the neonatal intensive care unit. Most of the babies there are on ventilators. And it's a very complex disease. And part of that, so Gina's reading my mind now, this is amazing. So this is one of those diseases where it's incredibly complex, because what you have is a baby that, when born extremely premature, so at our hospital, you'd see 21 to 22 weeks, so they're just past the halfway mark of pregnancy being delivered. These babies survive. They survive the initial period, and what the physicians are then trying to do is actually to help them ventilate, to help get their oxygen saturations up, while minimizing the extent of injury to those lungs. So you've got lungs that are incredibly premature. When the mum is at risk of delivering her baby prematurely, she gets a shot of antenatal steroids. And what that does is it forces those very premature lungs to turn into alveoli. There is the flip side to that, and that is alveolar stunting. So you force them to grow, they don't grow as they normally would, and they don't create that very complex structure that you need in the lungs for good gas exchange. So what you see in that, that top panel is the canalicular and the saccular stage, and that's round about where the lungs are when they start to, when they are at, you know, being born prematurely. They get ventilated, they've seen steroids. Some of these babies will have another shot of steroids or maybe more to try and increase that alveolar development while being at risk of alveolar stunting. And that bottom panel shows a lung histology from babies, one that died from non-BPD causes and one that died with BPD as the primary cause. And what you'll see is that the one that died from BPD, those lung sacs are what we refer to as simplified. So it's a very simple structure. There's not a lot of surface area for gas exchange. And those babies, in addition to BPD, once you have moderate to severe BPD, they are also at risk of developing pulmonary arterial hypertension, so cardiovascular diseases, as well as neurological diseases like cerebral palsy and necrotizing enterocolitis, which is referred to as the specter of death in a neonatal intensive care unit. So we're a little bit like, I hesitate to use this, but a little bit like COVID-19, where it starts off as a respiratory disease and that's what you're trying to manage, but really what you have is multiple organ damage. Next slide, please. So this cartoon here is a little bit of a summary of a review article that was written by colleagues, not by us, but it's a really nice review that talks about how complex the disease is. And when you're trying to develop an EV therapeutic or a cell-based therapeutic, you need to really understand your disease and understand how your therapeutic can influence the disease pathology, and in so doing, understand your mechanism of action. So Gina, there's a bunch of animations. I think if you just click through them. So as Gina's animating these, what we're showing here essentially is how over about a decade, we went about to try at the discovery stage at the bench with animal models, try to understand how the amniotic epithelial cells could modulate inflammation by interacting with specific cell types to get the outcome that we want, how they would activate endogenous stem progenitor cells, because while the lungs undergo alveolar stunting, they still possess endogenous stem progenitor cells. So there is still that opportunity to actually enable the lung to regenerate in those babies. And lungs that are exposed to high levels of oxygen, they do two things. One is that the cells will die over time in response to the oxidative stress, but also what happens as a result of high levels of oxygen is that the lungs actually start to downregulate the angiogenic signals. This is really critical to the pathophysiology of the disease, because during lung development, the development of new air sacs, so alveologenesis, is strongly tied with the development of new blood vessels, so angiogenesis. So when you knock off angiogenesis, lung development, the development of the air sacs are also compromised. So we're trying to address a number of different things here. And through that period of time, we were able to, by answering fundamental research questions, we could then understand the mechanism of action of the cells, and in so doing, the EVs later on. The other clinical release criteria that we have to think about with cells, and the same thing applies with EVs, is the sterility. So how would you go from a research lab level of tissue culture sterility to having a therapeutic that you can manufacture and release on demand? We never tested whether or not our cells were endotoxin free before going into our preclinical animals, whereas we had to with the clinical trial. So that was a bit of a learning curve for us. And not so much with the EVs, but with the cell-based therapies, we have to look at things like cell viability. You know, at what point do you decide that this batch does not actually meet the release criteria, and what do you do with them, having invested all that time and money? So on to the next slide. And I did also mention earlier on that I wanted to touch on selecting your animal model. So we have used a combination of small and large animal models in order to answer our question. And what we've done is teamed up with a group in Perth in Western Australia, and so Professor Jane Pillow is the lady that you see on the top left-hand corner with that little clipboard. So she's a neonatologist, and our own center, the Ritchie Centre, has a large team that works on ovine models for neonatal indications. So between our two sites in Melbourne and in Perth, we were able to have this sort of united effort to develop an animal model, a large animal model. So our small preclinical animal model gave us the ability to investigate things with molecular tools, because there are abundance of probes for, flow cytometry. If you wanted to look at microRNA signaling, you could do all of that with the mouse. But in terms of lung physiology, and bearing in mind that corticosteroids and development of surfactant for these babies was developed in sheep, that made sense to us to go back to a large animal model where those early discoveries were. The other cool thing about going to a large animal model like with the sheep is that we could deliver them prematurely. We could ventilate them and essentially treat them like they were babies, because the lung development in the ovine model is very well established. And a lot is known about how you can ventilate them injuriously versus gently. So you could almost vary it so that you have a severe BPD and a moderate BPD in a lamb that was delivered extremely prematurely or very prematurely. So this was one of the critical parts to our efforts. Gina. So what I've included here is the sample set from our first in human trial. So with all of that, about 10 years worth of discovery research and our preclinical studies, we then embarked on our first in human clinical trial. And in that, we had 6 babies included. The first 3 babies were mechanically ventilated, so they were intubated. The remaining 3 babies were on CPAP. And I just want to draw your attention to the gestational ages. So they were all severely premature, so 28 weeks of gestation or earlier when they were delivered. Their birth weights were all well under a kilo, except for baby number 4, that was pretty close to a kilo. 5 out of the 6 babies were males, and this is again quite typical for BPD. It's quite representative of the set. What you'll notice as well is that these babies don't just suffer from severe BPD. They have a lot of other complications that come along with being born so small and being ventilated. So most of them had pulmonary arterial hypertension, and some of them already had some indications of systemic hypertension, pulmonary hypertension. In this first in human trial, they were given cells at 1 million cells per kilogram, and all except the very first baby, we were able to hit that targeted dose. And if you just click onto it. So this is little baby Flea. So he was baby number 5, and that first picture was taken within days of him receiving cells, and second photo taken at his 2-year follow-up, and third picture about 2.5 years old when he came back, just to have one of his milestone meetings at the hospital. So the 2-year outcomes of this trial have now been published as well, and we are well on the way to understanding that, how well they're safe, how well they're tolerated in this patient cohort. Now the next slide basically shows you a series of snapshots that I've taken of our clinical trial protocol, and we've now moved to a first in human trial for patients with acute ischemic stroke. So in these patients, the cells are given within the first 24 hours of a confirmed infarct. And the second is, again, an early phase 1 clinical trial for patients with compensated liver cirrhosis. And we also have a dose escalation trial now for the prevention of BPD, because while we were going on with our clinical trial, that first clinical trial, what we were also seeing in the lab, because we were basically pursuing both continuing the discovery research and going on with the clinical trials, is found the earlier that you intervene, essentially the better the outcome, which is, I guess, no surprise. But we needed to have that data set, and here's a consideration if you're developing EV therapeutics. We needed that data set in order to convince ourselves, as well as the Human Ethics Committee, that not only are the cells well tolerated in this patient cohort, but we're able to deliver the cells safely. And now that we've done that, we can then go back to the Human Ethics Board to say, well, how about a preventative strategy where we can kind of predict which one of these babies are going to develop very severe BPD based on a risk estimator, and then go ahead and approach those, the parents of those babies. We do have a trial that's not, the protocol isn't published, but it's a trial that's just newly been registered late last year, and that's for perineal fistulas secondary to Crohn's disease. Next slide, please. So the current challenge that we have in these little babies is that they have, they're very small, so their blood vessels are very small as well. But while they are on a ventilator and exposed to high levels of oxidative stress, their pulmonary capillary bed is actually hyperactive, they're hyperresponsive. So passing cells through there, there's always the risk of pulmonary emboli. So that was one of the things that kind of tweaked our interest in pursuing EV therapeutics, because if you went with something that was far smaller than a cell, then you reduce the risk of pulmonary emboli in these kids. The other thing is, because they're so small and their circulating volume is so tiny, you need to have a very slow IV infusion rate. And this is not something that we've seen in the adult trials, and people haven't reported this in cell-based trials, even with children, because they have a much larger circulating volume. So little Emmanuel is the child, the first baby in our dose escalation trial, and you can see how tiny he is. So you can imagine his circulating volume isn't huge. And having that slow IV infusion means that you can physically see the cells settling in the IV line. So that raises questions around how do we deliver what we think is a therapeutic dose? How do we assess efficacy if you have to infuse the cells so slowly that the cells potentially settle in the lines? Another thing is with these patients, because of their vulnerability and the fragility of these patients, they need to come from, the cells need to come from CMV negative donors. And you can imagine that, you know, trying to, when you tighten the recruitment criteria for your donors as being CMV negative, that becomes incredibly tough to get enough cells going in. So, you know, these multiple of reasons then accumulate to kind of give us the incentive to try and develop an EV therapeutic. There are also secondary complications that are non-respiratory that you can kind of imagine, you know, if you had a therapeutic that could be delivered intranasally, for instance, in babies that are this young with the blood-brain barrier not fully formed, perhaps you could then access the brain through an inhaled formulation. Not just because you can doesn't mean you should, but that is an option, I guess, with EV-based trials. And increasingly over the years, we've had a number of international sites approach us to provide cells, and from personal experience, this is incredibly expensive, and there is no guarantee that the 90% viable cells that you ship across will be 90% viable when they arrive. And it also requires that every site that does your clinical recruitment actually needs to have the same ability to thaw, wash, and formulate the cells in the same sort of way. And this becomes very challenging as you increase the number of clinical sites to your clinical trial. On to the next slide. So we started off with an idea of cell-based therapies, and you know, this stuff goes back to 2005 was when we published our very first paper on these cells. And it was still in an era where people were thinking that cell-based therapies meant that cells would engraft. Those of you who've been in the regen med field for long enough probably do remember the time where pretty much you'd submit a paper and a reviewer will by and large ask you about the level of engraftment and how you showed the level of engraftment. And the story sort of started off there, and we, much like every other person in the field at the time, was looking towards that. And over time, we now know that, you know, engraftment has very little to do with efficacy, and that engraftment is not necessarily the be-all and end-all of a cell-based therapeutic. And that eventually evolved to the secretome, the paracrine effects, and EV-based therapies. So we were interested in that for, you know, the reasons that I already explained earlier. If you just animate the next, Virginia. So we were interested in a particle that was smaller compared to cells and address, you know, the cell settling sort of a phenomenon. We were interested in maybe immortalizing cell lines that from donors that were CMV negative, and just never having to have to deal with that issue again as part of our release criteria. You know, is the donor CMV negative? We were very keen for a simplified cold chain logistics. What might not be so obvious is that these babies can be incredibly unstable. So you may only have a short period of time to deliver an experimental therapeutic to assess for safety, because if you just stand next to their monitors, you'll see the numbers fluctuate every which way. And it's difficult for the physicians to then be assessing safety over a 24-hour period when so many things are going crazy. So if you have something that, you know, you have on hand that, you know, the baby's been stable for 2 hours now, the neonatologist believes it's safe to go ahead with therapy at this point in time, and then be able to do that rather than get the team together, you know, identify the cells, get them from liquid nitrogen, thaw, wash, formulate. That in itself could take 2 hours. We're very keen to have a cost-effective therapeutic. So neonatal ICUs are generally in large public hospitals. So, you know, thinking ahead, who would be paying for it? It would probably be the taxpayers. So could we create something that didn't further add to the cost divide or the wealth divide for those who can and can't afford regenerative medicine? We also were, of course, interested as scientists to have a product that was less variable. So we have, I'll talk to you about potency assays in a little bit, but we do see in our potency assays, some of the batches will just fail. The donors appear to be perfectly healthy, you know, healthy elective cesarean, gestation was at term, mom wasn't on any particular medication that should make any difference, and somehow the cells are just not potent in terms of, you know, T cell suppression, or they might pass everything, but they don't, the macrophages don't polarize in response to the cells, and then that batch of cells just doesn't get used. So we're very keen to have a cell-free therapeutic that was consistent across the board that we could manufacture en masse. And on to the next slide. So I thought I'd tell you about how our methodology evolved. This is how we prepared the cells for the preclinical assessment. This was probably about 5 years ago now that we started this. Learning from a lot of experts in the field at the time, you know, and working with the tools that we had. So we had previously published on conditioned media from the amniotic epithelial cells after 96-hour culture. And we thought, well, if 96-hour culture gives us the conditioned media effects, so the secreted effects, and the secreted components will contain EVs, then 96 hours is the way we go. So we had static cultures, we grew them we had the cells in the media for 96 hours, then we had the conditioned media we would then subject to ultracentrifugation. So the conditioned media, the media that we used at the time was a serum-free chemically defined media, because by that time, by 2015, we were already in clinical trials. So the only reason why we weren't using a media that contained FBS was because we had already had to go through that rather painful learning process. We were using an ultracentrifugation method because it was very common at the time. We had people who could teach us. However, it's not scalable. It's an open process, and by that I mean it's not within a device. You know, you've got lids that need to come on and opening and closing of lids, and it's got to go into a rotor. And because by 2015 we'd already been in the clinical trials, our minds were already, you know, if this works, how could we transfer this into a clean room? And so we weren't a big fan of it, but that was the tools that we had, so we went ahead with that. We did notice that from the same cell line, or even from the same batch of pooled conditioned media, there were operator variability or operator error. You know, is that a pellet? Is that not a pellet? I think that's about 1 ml that's left in the tube. I think a number of you could probably relate to this. At the time, we didn't have a device in our lab or in our institute that could reliably quantify EVs either. So what we did instead was most of our quantification was based on BCA assays. And I know that this is a bit of a contentious field, and not being an EV person primarily, I'm sure there are others who could argue it one way or the other, but that was the way we went about it. We did acknowledge that it only accounted for the protein cargo. So if our mechanism of action was based on microRNAs or non-coding RNAs or lipids, we were not capturing that, but that was the way that we were doing at the time. And we would go to La Trobe University or Melbourne Uni to use their nanosite. We would do Western blotting, FACS, ELISA, and EM. And so we had this whole battery of things, and it was incredibly slow for us to say yes or no. This is no longer the way we do it, but this is how we did it for the preclinical data set I'll show you in a bit. So in a quick summary, and we've published this, so this is just to quickly summarize what we found, is that we are able to isolate the EVs. For better or for worse, the EVs that come from the amniotic epithelial cells over the 96-hour culture period using the culture media that we do use is it's got quite a narrow size range, so 70 to 120 nanometers that we see on EM, and that's also seen on the nanosite. We find that they do package HLA-G, quite a fair bit of it. I mentioned this because the amniotic epithelial cells produce a lot of HLA-G, and that's one of the major immunomodulatory molecules involved in maternal fetal tolerance. So that to us was like, oh, that's pretty cool. So it probably will have the same immunomodulatory effects, we hope. The tetraspanins that they have are all of them, but by and large, you see one, not all three of them in the HAC-derived EVs. When we did a Western blot, we did see these so-called exosome markers, ALEX and TSG101. How relevant that is, I'm not entirely sure. We've done mass spec and pathway clustering and the enrichment of the proteins, and we've not done so much with the microRNA or small non-coding RNAs, but certainly with the mass spec, we show that the proteins kind of cluster around stem cell signaling, cytokine, chemokine signaling, integrin signaling, which all kind of make sense within the question that we're asking at the moment. So we went on, after being able to isolate the cells, one of the animal models that we went to was a preclinical model of BPD, but this time just a small animal model because we wanted a proof of concept. And also bearing in mind that at that point in time, we weren't able to isolate these cells, these EVs at any sort of scale, really. So what we do is at E16 of gestation, we exteriorize the uterine horn, so you can see all of the pups in their individual amniotic sacs there. We inject lipopolysaccharide into each one of those amniotic sacs, and through fetal breathing movements, it introduces the LPS into the lungs. So you get the first hit of antenatal inflammation that then starts to mimic BPD. The pups are then returned into the mother, stitched up, and then the mums deliver their pups at roundabout term that we indicate as day 0. And as soon as the pups are born, they're randomized either to 65% O2, so hyperoxia. So between the LPS and the hyperoxia, you basically get this 2-hit that gives you a very BPD-like kind of lung histology. So architecture changes, vascularization changes as well. Cardiovascular complications associated with BPD are also mimicked in this model. The inflammatory cytokines in the lungs kind of mimic what we've seen in babies with BPD. And at day 4, so while the pups have been exposed to 4 days worth of high levels of oxygen where lung inflammation is well and truly set in place, we then intervene with the EVs. And we give either 1 or 10 micrograms of EVs, either from term donors or preterm donors, because this is us going back to the drawing board and doing discovery work all over again. The question that I had at the time was, do the EVs work in this model? And does the gestational age of the donor work? We know that the cells don't work, but cells and their EVs, I don't think you can necessarily say if the cells don't work, the EVs won't. I wanted to answer that question, so that was the way we went about it. And on day 14, so the red arrows indicate when we culled them. So at day 14, we went and looked at lung histology. By week 4, what we would do is actually the mice, the animal house would actually wean the mice at that point, and we would take them from the high levels of oxygen to 21% room air and weaning them off the high levels of oxygen as well. And then at week 6, we would then do the physiological testing, such as lung function testing. And Dan Danju, the postdoc in our lab that's been doing this work, she's a medical imaging specialist and has a medical background. So she was very confident doing echocardiography on these fairly small mice. So we were able to tell if the lung function actually did correlate with the changes in lung architecture that we saw on day 14. So I'll just present a couple of bits of unpublished data in here. What we saw was there was an impact on alveolar simplification. So I'll talk you through this slide. So on the left-hand side, what we have is a tissue to airspace ratio that's graphed there in a quantitative manner. So the black bar shows our saline control. So these are healthy mice. For all intents and purposes, these are healthy mice. They've got lots of tissue to airspace. So it means that the lungs they retain that complex structure, healthy mice. When you expose them to the LPS and 65% O2, you get a loss of the tissue to airspace ratio. So you get alveolar simplification. And if you look at the lung histology on the right-hand side, you'll see that when you have that injury and you just give them a vehicle, you see that alveolar simplification. When you give the cells, you do get a restoration of that tissue to airspace ratio. You get a similar sort of an effect with the EVs from term cells, but you don't so much get an effect when you have EVs from the preterm cell. So it's looking like so far that the preterm EVs don't work quite as well as the term EVs. But then when you look at you can kind of see a trend that even the preterm EVs still maybe look a little bit better than just the saline alone, but this was not statistically significant. Next slide, please. So the next thing we wanted to look at, because as I mentioned earlier, well, you have alveolar simplification. These lungs actually have stem progenitor cells. So what we were quite keen was to find out, do you get a restoration of lung architecture because bronchoalveolar stem cells or endogenous stem cells within the lungs are being activated? So one of the ways that you can do this is actually by immunohistochemistry. So the cartoon on your hand side shows the sort of hierarchy and the different types of cells that you get in the lungs. The bronchoalveolar stem cells sort of sit at the terminal bronchioles. And if you like, the septal crests in that area then kind of bud out and they form the new alveoli. So if you can activate stem cells in those specific regions, you can then get new alveoli forming and thereby overcoming alveolar simplification. So we looked at the number of bronchoalveolar stem cells per terminal bronchiole. So on average, do you get more of these stem cells being activated? And on the left-hand side, you'll see the answer was no. It didn't matter if the EVs were from term or preterm cells. They just did not activate the bronchoalveolar stem cells in the same way that the cells did, which was very interesting because even though the end result was similar, the mechanism of which they achieved this is apparently quite different. So the next thing we looked at were the type 2 alveolar cells. So these are sort of like a facultative progenitor cell compartment. It's like the precursor of the type 1 alveolar cells. The type 1 alveolar cells are the ones that conduct gas exchange. They're the really thin ones that you see around the alveoli. And that we saw the term EVs could influence. So there are two potential ways that this could have happened, and we're still looking to try and understand that. It could be that the bronchoalveolar stem cells actually are turning into type 2 alveolar stem cells, and all we're doing is we're capturing it at a different point, that because on day 4 they get either the cells or the EVs, that maybe giving the EVs just triggers a different part of that continuum. The other thing is that it could be that they're acting through completely different pathways. So we're yet to try and understand that. So next slide, please. If you want to animate the next slide as well, thank you. So the next thing we wanted to look at was the impact on vascular muscularization. So this is where the blood vessels in the lungs actually get thickened. And the thickening around those blood vessels is with smooth muscle, and where you get pulmonary arterial hypertension, which is a very common complication in these babies. So the immunohistochemistry that you see on your right-hand side is alpha smooth muscle actin staining in red. And what you'll notice is that in the injury group, so in the BPD-like injury group, you get this thickening, the circle of red is very thick. That tends to go with the HACs. Is less so with the EVs from the term cells as well, but that is lost in the preterm EVs. So far it's looking like the term EVs perform to a similar manner as the HACs themselves. So on the left-hand side of this graph, what you're seeing here, what we've done is actually separated it into the very small blood vessels, so the ones that are 0 to 5 microns in diameter, and then the larger blood vessels, 50 microns onwards. And it doesn't seem to matter whether or not you're looking at the smaller or the larger vessels, that in both of these cases, it's the cells themselves or the EVs derived from term cells that actually are able to reduce this vascular muscularization. Next slide, please. So far it looks like it's the EVs from the term cells that are more beneficial. So that's the way we'll go in terms of developing our EV therapeutics. In all of these, we did IV administration, but we also did a comparison between IV and intratracheal. and both seem to be feasible for the respiratory applications. Maybe we've kept the tissues for looking at other organ damage, such as the brain. So we'll be interested to see if the IV or IT route actually have much of a difference. This is important for BPD in particular, because if an IV administration is the same, gives you the same efficacy as IT, you would go with an IV route simply because it avoids intubating a baby that isn't already intubated. So if a baby that's on CPAP and doing very well on CPAP, the last thing you want to do is take them off CPAP, intubate them, and then mechanically ventilate them while you deliver the cells, simply for, or the EVs, simply for the sake of that experimental therapeutic. It also starts to raise confounding factors when you try and tease out your safety data, because you've now intervened with what would be standard of care for that child. So the EVs from the HACs can also prevent experimental pulmonary arterial hypertension. I haven't had the chance to show you that, but certainly we'll be including in the paper that we're writing. So that's quite useful for us, because being able to prevent not just the primary disease, but the secondary cardiovascular effects from that disease will be, I guess, gives us an added sense of encouragement that we are going on the right path. So the next steps that we're looking to do is actually to now work on scaling up our production of the EVs, and going into large animal studies, because those lambs can actually be delivered not just at a similar rate of prematurity, but at fairly closer weights of the human baby. So knowing what kind of dosage, route of administration, frequency of dose will then allow us to design a clinical protocol that is feasible and actually geared towards collecting efficacy data. And we're also, so Tantan is now also working on bioprocessing methodology for scalable manufacturing, and with a view to transfer to a contract manufacturing organization, because we're simply not in the position to do GMP manufacturing of EVs at our site. So I thought I'd just talk you through some of the learnings and considerations now that we've gone through all of this, and what we've been able to quite confidently take across from cell-based therapies to EV-based therapies. So first off, it's really important to understand the underlying disease pathophysiology for the disease that you're looking to treat with your EVs. Next slide, please, Gina. So with bronchopulmonary dysplasia, as mentioned before, it's a complex multifactorial disease. Consider if the murine models that you maybe are working with, are they necessarily the best ones? Most of the physicians that we work with, actually all of the physicians that we work with, while they're interested in biomarkers, are really keen to see as well, you know, how, for instance, at a particular dose, at a certain degree of severity, is there a predictable time at which a responder would be able to be weaned off a ventilator? Have you got large animal models where those physiological findings have already been delivered and can be part of the dossier when you put them forward to the Human Ethics Committee? So if you don't have that, I would suggest reaching out to collaborators around the world that would have those sorts of models set up and are perhaps more geared up towards physiological studies than your own home institution may be. If you're looking at biodistribution, there's already a fair amount of criticism around lipid-based labeling of EVs that I know are very popular and very simple to use, but perhaps start thinking about labels and ligands that will take you through from preclinical assessment all the way through to clinical imaging. So that way you're using the same label in your preclinical studies all the way through to clinical trials. Look at the route and rate of administration. So you might need to, again, have a chat with the physicians that you want to work with to find out what makes the most sense for them. And of course, as early as you can, define your target product profile. So when you have your target product profile, you may actually find that you start to tweak your experiments in order to address those questions. Once you have your target product profile sort of on a spreadsheet, it'll start to tell you, you know, what other questions have I not answered? What should be the next most pressing experiment for me to design? So for the, I'm going to speak next about potency assays, but the primary factors that underlie the pathophysiology of our disease primarily are low angiogenesis, low alveologenesis because of alveolar stunting, and constant oxidative stress and inflammation due to the ventilation that these babies are subjected to. So it's with that in mind that we went on to design our potency assays. So the potency assays that you want to develop should necessarily reflect your mechanisms of action. So in terms of a clinical release criteria, not only is it safe by way of endotoxin-free and sterile, but does it do what I need it to do in order to have an efficacious outcome? So for us, developing the potency assays were around those key criteria. Does it reduce inflammation? And the methods using the cellular crosstalk that we have uncovered in our discovery phase. Does it induce angiogenesis and alveologenesis? And all of these were actually developed while we were at the discovery stage of our research. And also consider your critical raw materials. This was one of the things that I wish someone had told me early on. So the critical raw materials refer to any kind of material that will come into direct contact with your product. So anything from dishes to pipettes to the more obvious cell culture media and your growth supplements and your antibiotics. So what I mean by consider your critical raw materials was basically don't get stuck in a situation that we were, where we had optimized, we'd done the academic thing. We had optimized our methodology. We knew what worked and we didn't want to change it. And then one of our critical raw materials supplier had an issue with providing us with the material for 6 months. And we were too far invested by that point to then start thinking, well, what's plan B? So if you're not at that point yet, I would strongly recommend start thinking about that now and start doing those experiments to optimize it. So you've got plan B and C for all of your critical raw materials. And if you are still using FBS, consider moving to xeno-free. Start investigating on things like GMP grade equivalents for clinical manufacturing. A lot of these will then allow you to tech transfer it to a contract manufacturer more easily. And if your culture system requires you to use bioreactor systems, consider going towards a bioreactor system that has single-use enclosed disposables as well, because that just makes the tech transfer that much easier. If you have access to like an academic GMP manufacturing facility, that becomes a lot easier because you're not relying on the training of individual operators to deliver a GMP grade quality. You want to engineer the human error as much of it as possible. And of course, don't puzzle it out on your own. I spent a lot of time puzzling it out on my own because we were working with a cell type that nobody knew about. But I think with EV Therapeutics, there are a number of industry experts that could help just because of the interest in this growing field. And also considering your manufacturing process even at this academic discovery stage. So with that, what I mean is, even as an academic, you might think, well, I don't need to know anything about regulators because my job is to discover stuff. I would say that the reality check for that came to me fairly late, because I had thought that my job in clinical translation was to make the discovery. I would then give it to someone would translate that for me. What I found was that I had to meet with regulators. I was called into human ethics meetings, asking me questions around what do the regulators think about this, and can you cite to me chapter and verse what that means. So I think save yourself that agony and just bite the bullet very early on. Figure out where does your therapeutic, based on your process, actually place you in terms of a risk framework, for instance. Don't just consider your product to the animals, but consider what it's going to look like or how you envision it looking like when it goes to a patient. Is it in a bag? Is it pre-filled in a syringe? Is it in a vial that needs to be then resuspended? So that's what I mean by what does your fill, formulation, and finish process look like? Because you may find that you start having to interact with chemical engineers to kind of figure that out, and industry experts to try and figure what that process should look like in order to fulfill regulatory requirements. I would also suggest start planning on the stability of your product. So if your product had to end up in a ward that is in Western Australia and it's a 47-degree day and the air conditioning is not really working, what is the lifespan of that product in that environment? It's good to know that very early on, and it's good for you to be the person that informs the regulators of that, rather than having failed batches at the other end and being reported on those failed products by a clinical investigator, for instance. If you're going to freeze your product, will you use cryoprotectants? Consider if the cryoprotectants are FDA approved, if you're not going to wash them out or filter them out before going into the patient. So if the cryoprotectant's going into the patient at the same time, and are those cryoprotectants contraindicated in the population of your patient interest? So for instance, with the cells, the FDA does not have a guidance document on babies that are that premature. So we cannot safely administer cells with DMSO that's part of the formulation. You can do it with adults, and that is FDA approved, but in adults. So have a good sense of your patient group and if the cryoprotectants are going to be appropriate in that group or tolerated in that group. Of course, there's that lovely old nugget that keeps turning up. Do the producer cell types produce enough EVs for your therapeutic applications? And if the answer is, I struggle to produce enough just for my mice, then I suspect that, or I would actually strongly suggest that you start addressing that problem now, because that is not going to go away. And again, speak to the industry experts. There are ways around that. So start incorporating those very early on. Are you going to be changing your method of cell culture and EV isolation once you get to that scalability step? And have you verified potency and purity once you get to that purity step? Because once you change your methodology, you do have to verify that. And it's best that it's verified by you because you best know your product. And again, check in with the industry experts. There are ways around this, and there's no need for you to struggle alone. So what I'm gonna wrap up with are just some final points. And I guess the first thing is, who are your clinical champions for the disease you're interested in? Have you got them engaged? If you haven't, you should. Chances are you will, and this is the exciting thing, you will get to co-develop your clinical trial, because from my own experience, the clinicians don't just take your dossier or your research package and run off with it. They want you to be part of the journey, and they recognize that you're an expert in the area. So connect with them very early on, and then be prepared to work with them as a team. It's not like, okay, you just run the trial now and I'll just continue on my next project. You will be in that relationship with them for years to come. Part of what you want to be able to, your contribution to the clinical trial protocol will be things like, do I collect urine or serum or plasma or whole blood? How should they be collected? Because what are the biomarkers that I've found in our preclinical discovery stage? What sort of medical imaging should be used, and at what time point? The neonatologists, in my experience, weren't able to do that. So, we work hand in hand to kind of nut all of that out, and bringing in experts from international bodies where needed. If you work in an purely academic setting, and you don't have access to hospitals or physicians who are necessarily interested, one of the options is to actually engage a contract research organization. They do cost a fair bit, but they will be one of the enabling steps for you to enter the clinic if you're not in a translational environment as what we have been. And also, the manufacturing process for most people will be tech transferred to a contract manufacturing organization. I would suggest vetting a few, most of them by word of mouth. You can find out which ones are the better ones. They may not be in your home country even. You may have to go overseas to access a contract manufacturing organization. And they'll be the people that will manufacture it to the grade that will pass regulatory muster, that will be safe for the patients, but also they will take on board not just your protocols for manufacturing, but as I've listed here, the potency assays, your sterility test, and how you quantify it. So again, it's not like, here is my product, now make it. It will be very much working hand in hand with them, and engage with them earlier if you need the contract manufacturing organizations to actually develop some of those things for you. So I believe that's my last slide. I just need to, oh no. So these were my tips. So you've heard that fortune favors the bold, but what maybe you haven't heard is that it also favors the prepared. So prepare as much as you can really early, be really determined because you'll have plenty of roadblocks, and it costs a lot. So the sooner you can access funding, whether or not it's through commercialization or by reaching out to philanthropy or patient advocacy groups, do it. Even if it doesn't mean that you see funding for years yet, having the funding will help because trials cost a lot more than you think they will, and they will always run for longer than you think they will. So thanks everyone for listening. And just want to acknowledge all the various people in my lab, the funding bodies, and all the collaborators that have been involved. Cheers.
Hi, Rebecca. Thank you very much for a really interesting talk. Carolina is unfortunately still having some trouble with her internet connection, so I'm going to help with the questions section. So I think the first question we had was from, was it Layla? You had a couple of questions. Are you able to unmute yourself?
Yeah, I've just unmuted myself. Hello.
Hello. Hi, thank you so much for this presentation and for sharing your experience. It's truly wonderful work you've done. I'm not an expert in this field. I'm just like starting. And I had a couple of questions. I'm trying to find them again. First, about the vesicles that you ended up with. I was wondering if they were actually exosomes or also microvesicles, and like what technique did you finally use? Was it the differential centrifugation or something else?
So the preclinical study that I showed you, that data sets actually from using sequential ultracentrifugation, which I think is the method that most people start off with. Mm-hmm. We never had much luck with any of the precipitation methods. So we kind of stepped away from that very early on because it just didn't work for us. So that data set was with ultracentrifugation. Gina is now in the process of developing a tangential flow filtration combined with size exclusion chromatography on a like sort of medium scale. Tantan's now working on bioreactor development and large scale processing. So Tantan's really taken on like what we think would be the commercialization data set for manufacturing. I can't, unfortunately, as you would hope, I can't disclose everything because it's confidential at this point, but hopefully we'd be able to share that in later years. You did have a question about microvesicles. So based on sizes, we don't think we had a lot of microvesicles. Mm-hmm. I just hesitate with the whole exosome versus non-exosome and the heterogeneity of the EV thing. So what I can say is they are extracellular vesicles. They are of that size. They do have some of those exosome markers that people refer to in earlier publications. I would hate to put anyone offside by calling them exosomes or not calling them exosomes, but that's sort of the data package that we're putting towards the regulators. They are EVs, and these are the physical characteristics that we see.
Okay, thank you so much. Also wondered, actually, you said that you didn't use any serum for the growing of the cells, right?
Yeah.
Well, in the lab we used to use exosome depleted serum. Would that be an option potentially for you?
I think others here could probably comment on that better than we can, because we moved away from using serum in our culture media coming on 7 years now. So about 2013, we pivoted away from using FBS in our cell culture. So we're very happy with the yields that we get in comparison to MSCs, and Gina were the ones that did the math, so she can probably jump in here. We get something to like two magnitudes of order higher EVs from the amnion cells, the amniotic epithelial cells, compared to MSCs grown in a similar manner. So we're pretty happy with our yields.
That's great. And lastly, I think my last question was about the number of extracellular vesicles in an injection, and kind of like about like, sorry, the percentage that ends up in the lungs, and because I've read that it mainly ends up in the liver sometimes, so I was kind of wondering about that.
Yeah, so we don't see any effects in the liver with the BPD model. We have done liver cirrhosis, liver fibrosis experiments where the EVs are injected intravenously, and we do see effects in that model. We have not tried to quantify it so far. We've only just started working with a radiochemist to develop a radioligand where we can measure where the biodistribution is in real time, rather than relying on necessarily lipid-based stains, which we're hoping to move away from.
Okay.
So I should have an answer to that in about a year.
All right. Thank you so much for your answers, and best of luck with everything.
Thank you. Thanks.
So Rebecca, could you also make me a co-host, because it might make it a little bit easier for me to line up questioners. And then we had a question, which I guess you kind of covered there, from Eramina, wondering about the way that the EVs were administered. So was it just intravenous injections was the main way that you've been introducing them?
So far we've done it 3 ways: intravenous, intranasal, and intratracheal. And the intravenous route is the one that we would like to go for in general. So as you've seen with our cell-based clinical trials, we're across a number of clinical indications, mostly because of the interest from the physicians on our site. And we were initially thinking, you know, intratracheal will be really good because most of these babies have a trach tube in anyway, right? So we'll just put them in that way. Turns out to be not so much the point, because of the way medical practice has evolved. While we were busy at the bench, the neonatologists have been working on gentler ventilation strategies, and being able to get these babies on CPAP rather than a mechanical breathing tube, if you like, has been the way to actually protect these babies. So as a result, I think what we would like to do is actually go with IV, because that way they can remain off a ventilator for as long as possible. The time on ventilator actually increases their risk of developing more severe disease, not just respiratory disease, but also neurological disease. So while we've tried multiple routes of administration, at least for the BPD cohort, we would like to go with IV.
Excellent. So I think that covers perhaps your question as well, Faiza. Maybe you'd like to come in if there's anything else you'd like to ask. Okay, so then if we move on to Nawaz.
Hello.
Oh, Faiza's here. Okay, yeah.
Hello, thanks for a nice presentation. I just want to know if, can you hear me?
Yeah.
Oh, okay. If serum-free media utilization have any effects on your cell? As you know, there are some people saying that if you use serum-free media or EV depleted media, the proliferation of your cells can be affected. Did you have seen something like this?
So we haven't, for 2 reasons. One is that the HACs, the amniotic epithelial cells, don't proliferate much anyway. So the data that I presented as part of the background, so ourselves and a number of other groups around the world have shown that these cells will get to passage 5 if you're lucky. Generally, passage 3 is about as far as you can get them. They'll either senesce or they'll undergo epithelial mesenchymal transition, and they're no longer the cells that you started off with anyways. So passaging the cells was never really an interest for us. I have come across a number of papers, as you've mentioned, where, you know, moving away from media containing serum can compromise cell proliferation. I guess the challenge is always, how do you demonstrate that your media is, or the EVs are now free of animal protein, which will then, or animal substances, even viruses, that could then pose a safety risk. So that will be one of the questions that you have to address with the regulators. And that's not to say that, oh no, my cells completely rely on serum, there's no way I can translate this to the clinic. It does mean, however, that you have to be quite, well, not quite, incredibly fastidious when you approve the critical raw materials coming in contact with the cells. You would need to have additional levels of surveillance to ensure that every batch of EVs that is released is not contaminated with some kind of animal product that would have come from the FBS.
Thank you.
Pleasure.
Yes, great talk, Rebecca. Just a question regarding the potency assays. How did you establish the levels which tells you, yep, this is correct, oh, this is not correct?
So what we did was ultimately reverse engineer, if you like. You're an engineer, aren't you? So you like my term. I picked it just for you. So what we did was ultimately reverse engineer our potency assays. We had always kept vials, of those cell lines that we used in the animals. And we noticed that in some experiments they worked, some they didn't. And more likely it was a donor variability than the animals given, same animal house, same inbred strain, same colony of mice. So then we were able to, go back to our potency assays and work out which ones worked in vivo, which ones didn't, and at what point, is it 20% suppression or was it 30% suppression that really, where it falls off a cliff, for lack of a better phrasing. Yeah.
Fantastic. Thank you.